Hi everyone, welcome to Neurobiology at Providence College. I'm Joe DeGeorgis. In our first lecture, we reviewed a paper by Tom Reese and Morris Karnofsky in which they showed that endothelial cells that line the circulatory system of the central nervous system are held together in such a way that precludes horseradish peroxidase, a stain, from moving from the circulatory system into the brain. This work suggested that the endothelial cells and the tight connections between them form the blood-brain barrier. However, endothelial cells don't only line the circulatory system found within the brain, they also line the circulatory system found throughout the body, and scientists that were studying the endothelial cells couldn't see a structural difference between the connections of endothelial cells that line the circulatory system in the body, which allowed stain to pass from the circulatory system into the tissues, as shown by Ehrlich, as opposed to the endothelial cells that preclude HRP from entering the brain, as demonstrated by Reese and Karnofsky. This brought into question whether Reese and Karnofsky had identified the blood-brain barrier. In today's paper, Milton Brightman and Tom Reese addressed the issue of whether the connections between endothelial cells that line the circulatory system of the brain look the same as the connections of endothelial cells that exist in the circulatory system throughout the rest of the body. The title of today's paper is Junctions Between Intimately Opposed Cell Membranes in the Vertebrate Brain, and this was published in the Journal of Cell Biology in 1969. Let's start by drawing a diagram like the one we drew for Reese and Karnofsky. We said that there were endothelial cells that line the circulatory system of the blood vessels in the central nervous system in the brain. And we said that there were red blood cells, and they were black because they have an endogenous peroxidase. And HRP, horseradish peroxidase, also turns black when subjected to a series of chemicals. So we had HRP that was filling the internal space of the capillary, the lumen of the blood vessel, and Reese and Karnofsky showed that you could get HRP that came up in between some of the gaps between the endothelial cells, but at some point they stopped because the cells were so closely held together that the dye couldn't get through into the brain. Out here and out here. But the argument is that if we have endothelial cells that line blood vessels outside of the brain, where you have another tissue type like muscle, then the HRP or other stains, like the stain used by Ehrlich, could pass through into the muscle in this particular case, in this example, and stain the muscle tissue. So what is it about the endothelial cells that line the circulatory system of the body that allows the dye to leak out into the other tissue types and stain that tissue, versus the brain, which precludes both Ehrlich stain and HRP, in the case of Reese and Karnofsky, to flood into the brain and stain that tissue type? I mentioned earlier that other scientists that were looking at the connection between endothelial cells that line the blood vessels of the brain and those endothelial cells that line blood vessels throughout the rest of the body couldn't see a structural difference between the connections here and the connections here. And that didn't make sense How is it that the stain can leak out in this case, but is blocked in the case of the endothelial cells that line the blood vessels of the central nervous system? I mentioned in our last lecture on Reese and Karnofsky that after the HRP was administered through perfusion, the brain was dissected out and it was placed, immersed, we said, immersion, in a fixative. So we have our brain and it's in a solution of fixative, which preserves the tissue. We said after that process, it actually goes into another set of stains. And these stains stain the cell membrane, it stains proteins and other structures within the brain, within the tissue itself, and ultimately it gets, we said, dehydrated in ethanol, and then so there's no water left, we place it in ethanol, and then finally we infiltrate it with epoxy resin and then solidify the sample. So this is epoxy. So to start their experiments, Brightman and Reese focused on different types of stains that would stain the membranes and the proteins differentially to see whether changing the stains would change the appearance of the endothelial cells and other cell types, in hopes of ultimately finding a structural difference between the endothelial cells that line the circulatory system of the body versus the endothelial cells that line the circulatory system of brain tissue. To start their staining experiments, Brightman and Reese decided to start by using a different cell type than the endothelial cells. They started by using ependymal cells. These are specialized glia that line the cavities or ventricles of the brain. Scientists at the time thought that these cells were tightly held together by tight junctions. Reese and Brightman say in their paper, in the abstract, certain junctions between ependymal cells have heretofore, of course that means before, been regarded as tight, pentalaminar occlusions of the intracellular cleft. That is to say, previously scientists felt that ependymal cells were held together very tightly. If we look at figure 1 from Brightman and Reese, you can see an electron micrograph, that is a photograph taken with the electron microscope by Brightman and Reese. And in that they show the ependymal cells. And there are 4 cells. This is figure number 1. But you can see 4 cells, and the cells have cilia. So you can see cilia projecting at the surface of the cell. But you have 1 cell, and another cell, and a 3rd cell, and a 4th cell. So this is cell 1, cell 2, cell 3, and cell 4, and you can see the cilia projecting into the cavities of the brain, ventricles. And they show that there are regions, they just have an arrow here and it points to the membrane, and you can't see any gap between the membrane of cell number 3 and cell number 4. And that's the argument, that these 2 cells are held together very tightly to one another, that you can't see a gap between the 2 membranes. If we skip figure 2 for the time being and move to figure 3, they show 4 images. This is 3A, this is B, C, and D, and what they're looking at is this gap between the membrane of cell number 3 and cell number 4, for instance, and they are using their different staining techniques This one, they use osmium and potassium permanganate. Here they use potassium permanganate only. Here they use osmium only. And in panel C, they use osmium followed by uranyl acetate. And if they look at the sample stained with osmium and potassium permanganate, you see kind of a dark line here, and then you see a space, and then you see a dark line here, and you see a space, and you see a dark line here. If you use potassium permanganate only, you get the same pattern. And if you use osmium only, you get the same pattern. But if you stain this tissue with osmium followed by uranyl acetate, you see a line, a space, a line, a space, a line, a space, and a line. It's a little difficult to see, particularly if you have a printout, but if you look at the original manuscript, you can see this pattern. And the only difference, in all cases, they are looking at the space in between the two membranes of adjoining ependymal cells. So the sample is the same in all four cases, but the staining technique used is different. Now, we all know what the plasma membrane looks like. It's a lipid bilayer, of course, and you have the heads of the lipid and then the tails. And the tails, of course, are hydrophobic. They don't like water. The heads are hydrophilic. They're okay with water. And when the plasma membrane is formed, the heads face the outside of the cell and the inside of the cell, which are both aqueous. And the tails come together. So again, what we're looking at here is the coming together of the membranes for cells 3 and 4. Of course, this is flipped over, so it's really like this. You have cell 3 like this, and cell 4 like this. So this is the part that we're looking at. So back to our lipid bilayer. Of course, this is the bilayer of one cell, of cell 3. So it's the lipid bilayer of this particular cell. And then we're going to have the lipid bilayer of cell number 4. And so it's going to have phospholipid heads and the tails like this. And like this. So this is the cell membrane from cell 3, and this is the cell membrane from cell 4. And it turns out if you stain the tissue, the phospholipid heads turn black. So these phospholipid heads are black. And then it turns out that the tails don't take up the dye very well, so they are white, so you have a gap. And then you get the second line, which is black again, and then you get a line that's black, and then you get an area that's white, and then you get an area that's black again. So in this case, where they used osmium and potassium permanganate, these heads are stained in a manner that just give you one line. It just looks like one black line. So these two lines are fused together and you can't see a gap. So you see the top line, you see a space, you see the middle line, you see a space, and you see the bottom line. One line, space, one line, space, one line. So it looks like you have 1, 2, 3, 4, 5 different layers. And that's true if you stain it with osmium and potassium permanganate. It's true if you see it stained with just potassium permanganate or just osmium. But if you stain it with osmium followed by uranyl acetate, then you get a pattern where you have the heads stained black, then you have a space where the tails are, and then you have a line here, and then you have the heads, and then the second side of the lipid bilayer. So you have a space here, and then you have a line here, and then a space and a line here. So you have a 1, 2, 3, 4, 5, 6, 7 layered structure here. And you see the 7 layer structure in panel C stained with osmium and uranyl acetate. And again, the only difference here is the type of stain. The tissue is the same. But what they're trying to say is that you can see a gap right here if you stain with osmium and uranyl acetate. If you stain it in a different way, in conventional ways with just osmium and potassium permanganate or potassium permanganate or osmium only, then you see a 5-layered system. Even though there is literally a gap here, you can't see the gap if you stain it with these traditional stains, but if you stain it with the osmium uranyl acetate stain, you see that these are not held together tightly. They are not tight junction cell types. They are actually held together by gap junctions. So what this means is that scientists can be fooled in terms of what the structural configuration is, depending on what type of stain is used. So even though people thought that ependymal cells were held together tightly, it turns out they're not. They're held together loosely by gap junctions, not held together tightly by tight junctions. Now Brightman and Reese could use their new staining techniques to study the structural differences between endothelial cells that line the blood vessels of the central nervous system versus the endothelial cells that line the circulatory system of the rest of the body. So now Brightman and Reese went back and stained brain tissue and other sample types with the new staining protocol and looked at the endothelial cells and how they're held together. And you could have HRP here, and this is brain and brain. And then we have blood vessels that line other tissue types, like muscle. These are the red blood cells, of course, inside the lumen of the blood vessel. It's the muscle. And now, if we look at the interface between these two endothelial cells within the brain, in this case, and within muscle, you can see that in the case of these two endothelial cells, we end up seeing, whoops, sorry. A structure like this. So now we see with the new stain, in this case, you have a black band here for the heads of the first cell, of this cell, for instance. And then we have a white space which represents the tails. And then it turns out that you only see one band here because these two phospholipid heads, these two layers, are held tightly together by tight junctions. So you only see one line, then you see a gap, and then you see a line. So with the new stain, this sample has a 1, 2, 3, 4, 5 layer configuration. But if we look at the gap between these two cells, then the first cell here, this membrane right here, has the phospholipid heads like this, and then the tails and the heads. Like this, and when we stain it with the new staining technique, we get a line here for the phospholipid heads, we get a space for the tails, we get a dark line for these phospholipid heads, then there's a space here. So we get a white space, then we get a black line for the heads, we get a space for the tails, and we get a black line for these phospholipid heads. So now we have a 1, 2, 3, 4, 5, 6, 7 layered configuration. So now the new stain shows a structural difference between the way endothelial cells are held together in the blood vessels of the brain versus the blood vessels in the rest of the body. And that's why the dye can leak through in between these two cells, because these two cells are held together by gap junctions with a space, and in this configuration these two cells are held together tightly by tight junctions, and there's no space and there's no way for the dye to escape from the blood capillary into the brain. This is shown in many of the figures throughout the paper, and I'm not going to go through those specific figures. I'm going to leave this as the schematic here, but I'd like to say one more thing about how these cells are held together. So in this case, with these cells being held together by tight junctions, the cells are, are held together like this. So it's like you took a group of balloons and you compressed them all together and then glued the seam between the balloons. So there's no way liquid can get through, the stain can't get through, because the tight junctions are forming a belt all the way around the cell like this, and they form a sheet, and then the sheet of balloons wraps all the way around the inside to line the blood vessel. In this case, the cells are held together by gap junctions. Oh, I didn't draw that very well. With a little gap here like this. And there's spaces for the stain to move through the cells in all directions. In this case, in this configuration, from the inside of the capillary to the outside and back. So through this approach, Karnofsky, Brightman, and Reese showed that the blood-brain barrier is formed by tightly associated endothelial cells lining the capillaries of blood vessels in the brain. Okay, well, these are two of my favorite papers, and as we mentioned earlier, the blood-brain barrier is extremely important, particularly in medicine, because it's difficult at times to get medicines across the barrier to treat neurological diseases and disorders. See you next time.